Hello, good morning, and welcome to day 2 of the Oregon Wine Industry Symposium Viticulture Sessions. Today, Dr. Marcus Keller is going to speak to us about the general growth and function of grapevines through the winter months. This is a great introduction or refresher into the basic growth and physiology of the grapevine. This session will set the stage for our second talk today on sap flow pruning and trunk disease. Dr. Marcus Keller is the Chateau Ste. Michelle Distinguished Professor of Viticulture at Washington State University's Irrigated Agriculture Research and Extension Center. He received his Master's of Science in Plant Science and his PhD in Natural Science from the Swiss Federal Institute of Technology in Zurich, and he has conducted viticulture research and taught in North and South America, Europe, and Australia. His research focuses on environmental factors and management practices as they influence crop physiology of wine and juice grapes. He is the author of the textbook, The Science of Grapevines, one of my favorite grapevine textbooks. Actually, it's right here, which is packed with important information about grapevine growth and physiology. He currently serves as the science editor for the 2 journals of American Society for Enology and Viticulture. As Dr. Keller speaks today, please utilize the Q&A button at the bottom of your screen to ask him any questions that might come up. Enjoy the talk.
Hi from snowy Washington, everybody. It's not actually quite as snowy as it looks like, but like that behind me. This is from a couple of years ago. But I've been asked to tell you a little bit about winter basics on grapevine biology. So let's get right to that. When we look at growth cycles of grapevines, sometimes it helps to remember that when you're in the tropics, or you grow grapes in the tropics, they actually behave as evergreens. They never drop their leaves. They just continue growing, continuously produce fruit. But when you grow grapes in temperate climates like Oregon, Washington, the US in general, they behave as plants that alternate their growing cycle with the dormancy cycle. So they grow during the growing season, and then they go dormant during the winter. And that's mostly because green plant parts, leaves, flowers, fruit, and so on are generally killed at about 25 to 27 degrees Fahrenheit. In other words, dormancy simply serves to ensure survival during the winter. The leaves have to be dropped off, the buds have to go dormant, the plant has to prepare for the winter so it can survive for the next growing season. You can see that in the picture down here, and I'm going to just change my pointer to a laser pointer here. You can see this vineyard. This is in Washington, where we had an early fall frost, and the plants at the bottom have lost all their leaves, and only at the top, where it was a little bit warmer, they were protected here by a wind machine, the leaves actually survived. So this is the reason that grapes grow dormant. They have to survive the winter. Before I can really talk about dormancy, I have to bring up a couple of terms that many of you are probably familiar with, but maybe not quite all of you. Those terms, I will be using them throughout the presentation. They're xylem and phloem. The xylem here indicated with an X in the interior of a cane. This is a cross section of a grapevine cane. The xylem is essentially the wood of the cane or of the shoot or of the trunk, the roots. The xylem is composed of dead cells. These hollow cells that you see here looks a bit like a filter or a screen. These holes are vessels. They're xylem vessels. They're dead, and they're transporting water and nutrients, mineral nutrients, up from the roots into the leaves, all the parts of the shoot. So this is in the interior. On the exterior of the shoot or the cane, you have the phloem, the bark. The phloem is always in the bark. The phloem is composed of live cells. We call them sieve cells, and they're assembled into sieve tubes. And they transport water as well, and nutrients such as sugar and amino acids and so on from the leaves to the various sinks of the plant. Sinks are like flowers, shoot tips, the trunk and the roots and so on. Something else is that the xylem is always under negative pressure. The xylem is under tension. It's the water that evaporates from the leaves, from the surface of the leaves, that pulls the water up from the soil and then up through the plant. The phloem, on the other hand, which often runs in the opposite direction, is under positive pressure. So the leaves pressurize the phloem and then push water and nutrients down or over into those different other plant parts. So xylem and phloem, I will be mentioning them throughout this presentation. This is the same cross-section of a cane. When I talk about dormancy, I have to start with maybe a misconception or stop a misconception that is often uttered. I used to even teach this to my students, and that is that brown canes are lignified and green shoots are not. That is nonsense. For all we know, that is not how it happens. Lignification actually starts very early during growth, and it happens continuously. A shoot, even as it starts growing right after bud break, it produces xylem. The vascular cambium is this sleeve of stem cells right between the xylem and the phloem, all around the shoot. It also occurs in the leaves and in the roots. So this vascular cambium produces xylem cells towards the interior and new phloem cells on the exterior. So the shoot grows, it gains in diameter, it grows thicker. As the shoot grows, during the entire growing season, that cambium produces xylem cells, and those xylem cells have to be lignified. Their cell walls have to be lignified, reinforced like reinforced concrete, steel reinforced concrete, in order for those cells to withstand the negative pressure that occurs in the xylem, so that those cells can transport water after they die. So lignified xylem cells are dead cells that transport water. That means that lignification process starts very early, right after bud break, and it continuously continues as the shoot produces more xylem cells. Has nothing to do with shoot browning. When we look at this browning, what you can see over here on the right-hand side, the shoot turns brown in the mid-season around the time of veraison, or roughly that time, turns brown from the base up. That is actually dead cells that we're seeing. And it happens because within the phloem, on the outside of the phloem, we have a second cambium that forms. It's called the cork cambium. That cork cambium forms cork cells to the outside. And those cork cells, along with the cambium and some other cells that are produced, is what we call the periderm, or the outer bark. So that we now distinguish between the phloem in the inner bark. You can see this, all these green, dark green tissues here on the outside of the xylem. This is phloem. And then we have this gap where we have outer bark forming. And as that bark forms, it is cut off from the interior of the shoot. These suberized cells or cork cells are cut off and they die. When a cell dies, it releases phenolics. Phenolics oxidize, phenolic compounds oxidize, just like what you see in a wine or in a juice. Oxidizing phenolics turn brown. So in nature, when you see brown on a plant, that usually indicates dead cells. So the browning that we see on these shoots had nothing to do with lignification. Lignification happens in the interior of the shoot. Lignin is colorless, you can't see it. But when the cork cambium forms these brown cells, these cork cells on the outside, approximately from mid-season onward, those cells die, they're cut off from the interior of the shoot, they oxidize, and then they turn brown. So that's the distinction between brown periderm and lignified xylem on the interior. So now the shoot has formed periderm. And this is way before winter. This happens in about midsummer to late summer. This periderm is brown. The shoot turns into a cane, and that cork that the periderm produces serves to insulate the shoot. Obviously, some of you have heard of cork oaks. You use cork for your wine bottles from the cork from the periderm of the cork oak. Same thing. that is used for insulation. It's also used for protection against wounding, against water loss, against the entry of pathogens. So a very solid outer surface of the shoot and the trunk and the roots. Now, eventually, we don't know exactly when that happens, but gradually, as the periderm has now formed, the cambium, the vascular cambium that is between the xylem and the phloem, that cambium goes dormant, which means it just arrests cell division. There's no new growth, no new xylem, no new phloem production during the winter. So growth stops. Then, surely, a little bit later, the leaves will start to fall off. But before they do, they recycle their nutrients. Grapevines, like other plants, are very economical plants. They don't just waste compounds that they have previously produced. So they will recycle their nutrients back to the canes, back to the trunk and the roots. I tried this. I looked at some leaves on the outside of the canopy and leaves that were just falling off on the inside of the canopy. The difference is about 75% carbon, 90%, up to 90% of the nitrogen, and about 85% of the phosphorus are recycled out of those leaves and put back into the plant for overwintering. This is one reason that when you have an early fall frost, you can sometimes run into nutrient deficiency issues in the next growing season because the plant has not had time to resorb or recycle those nutrients back from the leaves into the permanent parts of the plant for the next growing season. So those nutrients are now back, and within a few days, there is abscisic acid being produced by the plant, ABA, the dormancy hormone, and ABA triggers sealing of the phloem. It triggers production of a compound, a carbohydrate compound called callose, and that callose seals the phloem. So that phloem flow stops in the canes and the trunk. Strangely enough, this does not seem to be happening in the roots. It's only in the canes, only in the above ground plant parts that the phloem is actually sealed before the winter. And this happens just a few days after leaf fall. In the spring, about the time of bud swell, bud break, that callose is removed from the phloem. That callose that has blocked the phloem previously is now removed very quickly within just a few days, and the phloem resumes its functionality to support the new growth in spring. This resumption of phloem flow or phloem function occurs very quickly across the entire plant, almost simultaneously in the canes and in the trunk. At the same time, during bud swell in spring, auxin, the growth hormone auxin, is produced by the shoot tips that are contained in those swelling buds and are starting to grow and divide. That auxin flows down away from the bud into the cane and down through the plant all the way into the roots. As that auxin flows down in the cane, it reactivates the cambium. Cambium resumes growth, it resumes cell division, it starts producing new phloem to the outside, new xylem towards the inside. This process is different from the callose removal in the old phloem that we have seen. The callose removal, the restoration of phloem function, happens very quickly across the entire plant within just a few days. The cambium reactivation, the new cell division cycle, doesn't start until later, and it progresses much more slowly. It starts first just below the bud that releases auxin, and then eventually in the trunk, and weeks later only in the roots. So the roots do not start growing until several weeks after the buds are starting to grow out. So I've talked quite a bit about canes and trunks, but really when we think dormancy, we mostly think about buds. So I need to tell you a little bit about what a bud is. And most of you probably know this, but there's some terminology that is kind of important. Remember that a bud contains a compressed shoot. What you can see here on in the picture on the upper right-hand side is a typical compressed, fully formed, preformed shoot complete with 8 to 12 leaves. You can see these leaves or leaf primordia sticking up here as these green flat organs that we can see here. And these are embedded in these brown is dead again, protective scales. Scales like the periderm on the shoot or on the cane. Scales serve for protection, for insulation and protection against pathogens and wounding. Grapevine buds are quite resistant to physical damage, to mechanical damage, as opposed to buds of some other plant species. And this is one reason that hail doesn't hurt next year's crop nearly as much as it, or that hail hurts crop not nearly as much as in some other plant species. So we have these fully formed compressed shoots, complete with leaves and cluster primordia as well. You can see one cluster primordium sticking up here to the side on the right-hand side. There might be another one here, but this could also be a tendril that is already preformed. This is not always completely clear. So these buds are on the shoot, in the leaf axil of the shoot. You can see the leaf scar. This is a dormant bud down here. The leaf scar off to the side, the leaf from last year. This down here on the right-hand side, on the left of the bud, is a former lateral shoot from last year, and then the bud, the compound bud, sits up here. We distinguish, I just mentioned compound bud, but we really distinguish mostly between prompt bud, a prompt bud, and a dormant bud. A prompt bud is in the leaf axil of the current year shoot. Usually that gives rise to laterals during the current growing season. The dormant bud sits in the axil of a prompt bud. So in the first leaf axil of a prompt bud, the dormant bud is formed, and that is the backup system for next year's growth. The compound buds are what gives rise to next year's shoots and crop. Those dormant buds are, like I said, compound buds because they comprise 3 buds. So we have the primary bud in the center, we have a secondary and a tertiary bud off to the sides. The primary one is usually the most fruitful, secondary bud is less fruitful, the tertiary bud typically not fruitful, with rare exceptions. And so we have this 1, 2, 3 system that essentially serves as a canopy backup system. You can see down here in the lower left, we have this typical compound bud with the 3 bud structure, the primary one in the middle, secondary and tertiary buds off to the side. The primary bud is the one that usually grows in the next growing season. These secondary and tertiary buds typically do not grow out unless, like in this case, the primary bud has been damaged. So here we have a case of winter kill or winter damage that leads to outgrowth of those secondary and tertiary buds. Then we have another kind of bud that we call the latent bud. Latent buds are former dormant buds that remain dormant. They do not grow out in the following growing season, but they could theoretically grow out even up to 30 years and more later. Here is an example. I took this picture back in Australia on a vine that was about 40 years old. And this is a dormant latent bud that finally grew out after they had cut off the trunk. So you can use this, for example, during pruning, if you want to renew a cordon that has gone bare, you can cut off all of the buds, the compound buds. You can essentially disbud the cordon. And very often you will find one of these latent buds or several of these latent buds will grow out, and you can re-establish new spur positions on the cordon. So now we're finally getting into these the dormancy terminology. We are supposed to talk about dormancy after all, since we're talking about the winter, the basics of winter. The definition of dormancy is that no growth occurs, complete shutdown of growth, no cell division, no cell expansion, no growth, but the basic metabolism inside the plant has to continue. The tissue has to stay alive. So there is respiration, there is conversion of starch into sugar and back. We will look at this into a little bit more detail later. But just remember that during dormancy, there is no growth, but there is still some metabolism going on. We can distinguish between three different types of dormancy. First, paradormancy or pre-dormancy. During paradormancy, bud break is suppressed by auxin that is generated by the growing shoot tip. So the growing shoot tip ensures that there's not too much competition with its own growth, and it suppresses bud break down the shoot, for example, of laterals for quite some time. So this is paradormancy. Buds do not grow out as long as the shoot is actively growing. Then we have endodormancy. Paradormancy gradually transitions into endodormancy. Endodormancy is what we call true dormancy, or sometimes we just call it dormancy. During endodormancy, bud break is suppressed by the bud itself, by something that occurs inside the bud. We are not exactly sure what causes endodormancy in many cases, but we do know that ABA, again, is involved. The dormancy hormone abscisic acid is produced by the bud itself and keeps that bud from growing out. Later, the bud transitions to ecodormancy or post-dormancy. This is an imposed dormancy, imposed by the environment, and bud break is simply suppressed by low temperatures. Now, at the same time, as the bud goes endodormant, it starts to accumulate, to measure temperature, and it accumulates what we call chilling hours or chilling units, or it's simply chilling. So the bud measures low temperature. We think it's somewhere in the range 30 to 60 degrees Fahrenheit. Once the bud, and this is variety specific, you can see over here in the graph, this is Cabernet Sauvignon and Chardonnay, 2 years. Graduate student of ours, Diana Zapata, did this work. That chilling accumulation happens gradually. The bud measures the temperature. As the bud is exposed to cool temperature, that temperature will eventually lift or release dormancy, endodormancy. And the bud transitions from endodormancy to ecodormancy. Once a bud is endodormant, once a bud is ecodormant, its bud break is simply suppressed by low temperatures. So first, the bud uses low temperatures to accumulate chilling during endodormancy. That low temperature will then lift the dormancy, will enable the bud to break, but at the same time, that low temperature keeps the bud from breaking. So it becomes ecodormant, and only when warm temperatures around 50 degrees Fahrenheit or higher return in spring, then does the bud grow out. Or so we think. Just before Christmas last year, 2020, I got an email from Napa, California, with some really strange pictures. Here we have bud break on the 21st of December in 2020. It turns out that the bud break happened on vines that had been very close to wildfires in late September during the last wildfire season in California. We do know from very old French research that heat can lift dormancy as well. So if we go to the two extremes of temperature, cool temperature, chilling, will lift dormancy. But at the other extreme, really, really warm temperatures, we think in the 105 to 120 degree Fahrenheit range, will also lift dormancy. That is a heat shock, a shock that the plant undergoes due to excessive heat, and then bud break is enabled. So dormancy is lifted. So when you play with fire, with wildfires, this can be an unwelcome Christmas gift that you can see, especially during a mild winter. The buds that have been exposed to these high temperatures close to wildfires, you will eventually break. And of course, you lose your crop if it gets cold again. So both low and very high temperatures will lift dormancy and make the bud transition from endodormancy to ecodormancy. So when does this all happen? Usually we think that endodormancy in our climate. Pacific Northwest starts around the beginning of September and lasts roughly to the end of October, early November. Just a couple of comparisons though. So we know that endodormancy starts approximately at the same time as the grapes undergo veraison. But that can be 4 to 8 weeks after shoot browning, after the browning of the shoot that I've told you previously about. So I used to think that a brown bud is a dormant bud, that a green bud is a paradormant bud or a bud that can grow out at any time. And a brown bud is an endodormant bud or ecodormant bud. It cannot grow out until it has accumulated chilling. It turns out, when Hector Camargo did this work, he was another graduate student in our program a few years back. Turns out that is not actually the case. You can have brown buds that are not yet endodormant. So this shoot browning and veraison are not really tied in terms of timing to the entry of endodormancy. We know that veraison is mostly driven by temperature. This is one of the problems of the current climate change. As it gets warmer, veraison occurs earlier and earlier, so that ripening occurs at higher temperatures. Not very desirable, right? But it also means that veraison, the timing of veraison is variable. It depends on the temperature. The timing of dormancy, on the other hand, is driven by day length. In our climate, this is about 13 hours day length. So early September, late August, early September is when our buds typically go dormant. That, of course, is a stable cue. So it doesn't matter what you do to the vines, whether you crop them really heavily or you pull the crop off very early or whatever you do, the vine is able to perceive the length of the day and then tell its buds to go dormant at the same time of year every year. We have here Chardonnay and Cabernet Sauvignon again over 4 years, and you can see that veraison is variable, varies by about a month or more from one year to another, but the onset of dormancy varies only by just a few days. So there's probably a minor temperature effect as well, but the major driver is day length. That just means that neither veraison nor shoot browning are really related to dormancy. Dormancy is entered at a specific day length. Then you have chilling. Chilling accumulation, of course, is a pure temperature effect. That means that the time that endodormancy is lifted, or dormancy is released, as we call it, is also variable, because chilling depends on the temperature history after the onset of dormancy. So that can be, we can have a warm fall, it takes longer for chilling to accumulate, or we can have a very cool early fall and chilling happens very quickly. So the release of dormancy varies in terms of time as well. And then the bud is able to break, but it will not break until it's warm enough. And that means that the time of bud break also can be very variable. We can have an early spring, a late spring. Variations of up to a month, as you can see over here, are not unusual. So we can have one year bud break occurs in April, another year occurs a month later in May, just depending on the temperature. So the timing of endodormancy, the onset of endodormancy is stable, driven by day length. The time of veraison is variable. We don't know so much about shoot browning. We think that is variable as well. But we do know that the end of dormancy and the time of bud break are also variable, driven by temperature. Dormancy is linked to cold hardiness. I'm not going to talk much about cold hardiness during this presentation. Maybe we can do this some other time. But something interesting occurs during dormancy. We used this red food dye and fed it to canes or shoots at different times of the growing season. And then we were interested to see how much of that dye would end up in the bud. In other words, is the bud connected to the shoot or not? It turns out that in a green shoot, in a paradormant shoot, the dye would move into the bud. This is not a paradormant shoot here, but you would get the same picture. that dye would move into the bud. You can see that red staining in the leaves, even inside the bud and all the way to the tip of the bud. As soon as the bud goes dormant, it disconnects itself from the shoot to protect itself from water flow during the winter so that it doesn't freeze when the water freezes inside the plant. So you can see that in a dormant bud, the bud is entirely green. There's no red dye at all in the bud. The bud is completely disconnected from the shoot, and that enables the bud to dehydrate. During dormancy, you can see the dormancy period here is this gray bar. The bud water content drops during dormancy from about 80% water down to about 45% water. So 80% to 45% water content during dormancy, the bud dehydrates. That water content then stays roughly constant around 40 to 45% all through winter, all the way to bud swell. And I will talk to you a little bit later about how this happens, but during just a few days during bud swell, as the bud prepares to come out and then grow again, the water content increases from 45 to 80% over a very short period. So dehydration during fall, during dormancy, low water content during winter, quick rehydration, the water content goes up as the bud comes out and is reconnected here during bud swell. That reconnection is shown here as dye firstly accumulating at the base of the bud and then just shooting into the bud as the dye removes all those inhibitions and reconnects with the shoot. The short of this is that a bud needs to be dehydrated so that it can acclimate to cold conditions. So in other words, dormancy is a prerequisite for cold acclimation, for cold hardiness. You can see in this graph up here, obviously the dormancy period. We've talked about the water content changing. The black and the red line are the cold hardiness of the bud. And we measure cold hardiness as LT50. You can see that over here, but LT50, that means the lethal temperature for 50% of the buds. So that would be the temperature that would kill about half of the bud population in a vineyard. You can see that during dormancy, there's not very much happening. The bud sits there. Maybe it acclimates a little bit, but once dormancy is lifted, the bud suddenly becomes much more responsive to temperature, both up and down. As it cools down, it gets colder in the winter, the bud is able to acclimate. It hardens off. It gains maximum cold hardiness. And as it gets warmer again at the end of the winter and into spring, that hardiness is lost again. So an ecodormant bud is responsive to temperature. An endodormant bud, much less so. But you have to go through that endodormancy process for the bud to be able to really acclimate to cold. What else happens in the bud? And I told you earlier on that dormancy means stop of growth, but there's still continued metabolism. Well, one of those metabolisms is that during paradormancy, early on, before leaf fall, you can see on the left-hand side in this picture here, starch accumulates in the canes and in the buds and in the trunk and roots as well. Starch accumulates. Starch is a storage carbohydrate. Now we're getting into cold temperatures. The bud becomes dormant and starch is gradually transformed into sugar. You can see that this black line up here is the total carbohydrates in the buds, and that remains approximately constant during the winter. Down here we can see the white or empty dots are starch. They accumulate, and then as the bud starts going through dehydration, the leaves fall off, starch is gradually converted into sugar. Initially, not very much, mostly starch accumulating, and then the starch content declines as the temperature gets colder and the sugar content increases up to a peak where we reach the minimum temperature, and then things switch around again. Sugar is converted back into starch. You can see that on this picture here as well. as the amount of starch, this is backwards in some ways, as the amount of starch decreases, the amount of sugar over here in red increases. So we have high starch, low sugar. It gets colder in the winter. We have low starch and high sugar. And at the same time, this correlates with the degree of cold hardiness. So sugar serves to lower the freezing point of the bud. We have cold acclimation. That means that sugar is necessary to enable buds to cold acclimate. They cannot cold acclimate with starch. Starch needs to be converted into sugar. Starch is for storage. Sugar is for cold hardiness. But now we have a problem. Sugar is also osmotically active. If it rains or if it gets warm and it rains, that sugar will attract water from the outside and the bud will burst. It will not break. It will literally explode. It will burst from the water that it cools in because the bud has too much sugar. So as it warms up again, that sugar is quickly converted back into starch. And that is that continued metabolism during winter, that constant conversion of sugar into starch and starch into sugar, depending on the temperature. The lower the temperature, the more sugar accumulates. The warmer it gets in the winter again, the more of the sugar is converted back into starch to prevent that bud from flooding and from bursting. This is one main reason. This is the last thing I will say about cold hardiness. This graph, you see the black line that shows this LT50 again, 50% of the bud population will die. The bud acclimates, you get maximum level of cold hardiness. And you can see as the temperature decreases, the blue field above, the temperature drops, the bud gains hardiness, remains hardy. As the temperature warms up, even in early January, the buds lose hardiness. They cannot hang on to that sugar that lowers the freezing point. You have to convert it back into starch. So there is this constant up and down in the sugar-starch ratio. And at the same time, there's this constant up and down, as you can see here, of the cold hardiness level of the buds, as well as of the bark and the wood, or the phloem and the xylem. But you can see that the phloem is much less responsive to temperature, is also less hardy than the buds. And the xylem is usually the hardiest part of the plant, but it too responds to temperature fluctuations because of this sugar-starch interconversion. So this happens throughout the winter, and it happens because sugar, yes, it lowers the freezing point, but it also attracts water into the bud. So as it gets warm and moist, the bud has to convert sugar back into starch, and it comes at the cost of a loss in hardiness. So the worst possible combination you can have in the winter is a long, warm period, a warm spell, followed by a sudden cold snap. When we have these Arctic air fluxes from Antarctica that happen sometimes in our climate, that suddenly happen overnight when we have these very strong drops in temperature. If they follow a warm period, that is when we get cold injury on our buds and on our plants. This is enough about cold hardiness, except I need to mention the roots. Roots are typically concentrated in top 2 to 3 feet of the soil. It doesn't matter what terroir adherents tell us, whether roots want to go down and tap into the rocks underneath. The vast majority of the roots are usually in the top 2 to 3 feet of the soil. One very strange thing that I didn't know until recently is that roots do not go dormant in the winter. They also do not seem to be able to have that starch and sugar conversion, so they cannot cold acclimate. They are neither dormant nor can they cold acclimate. In fact, in places like California, the southern US, roots can continue to grow through the winter, using up carbon or carbohydrate reserves. That is a main reason that Vitis vinifera, typical wine grape roots, are damaged at 21 degrees, around 21 degrees Fahrenheit. Rootstocks, which are from American species of grapes, which are cold hardier, don't die until about 14 to 18 degrees Fahrenheit. So grafted vines, at least underground, are actually cold hardier than our own rooted vinifera vines. One problem with root damage, and I'll talk about this in the next slide, they cannot generate the root pressure. So they go through an uneven bud break and uneven shoot growth. And this is why you have this uneven shoot growth on vines that have suffered root damage that looks like bud damage, but in fact, it is because the roots have been damaged in the cold. And that's because very often in our climate, the soil tends to be dry in the winter. Temperature fluctuates more in dry soil than in wet soil. And so roots can get damaged at relatively high temperatures. This is the reason that we tend to irrigate in the fall. We often irrigate, if we could, we would irrigate in the winter. We are now irrigating in Australia and Argentina in the winter because the winter is too dry in those climates nowadays. This is another reason that we plant deep. We want to plant down to 12 to 18 inches to protect the roots from cold temperature, from fluctuations in temperature. So I've mentioned root pressure. Root pressure is what leads to bleeding or sap flow typically in spring. So sap flow, the exudation of xylem sap from pruning wounds, you can have an example over here, varies between 0.1 to 1 liters per day, and it occurs at a maximum around the time of bud break. This sap flow that you're seeing from these vines is a result of root pressure. And the roots can pressurize the trunk base to about 2 to 3 bar. That is about the same pressure as what you have in your vehicle tires, in your pickup tires. So a fairly high pressure, and is generated by roots remobilizing nutrients, sugar, amino acids, potassium, other mineral nutrients from the wood, from the xylem, and they pump that into the xylem, into the interior of these dead cells, and increase the osmotic pressure, just like we've talked about with the bud and sugar. The osmotic pressure increases, and that attracts or pulls in water from the surroundings. So the osmotic pressure pulls water into the roots and then pushes that water up the trunk. So remember when I said initially that the xylem is always under negative pressure? I lied a little bit. This is the one case where the xylem is actually under positive pressure. It is pressurized because there are no leaves on the plant. They cannot pull water up. So that water has to be pushed up the plant in order to push out air bubbles from the xylem. When the water freezes in the winter, it pushes out the air, and that air does not go back into solution when the water thaws again. So it has to be pushed out. And many of you have probably noticed in bleeding sap, you can often see foam or air bubbles coming out of the plant, of the xylem. And that is the main function of the bleeding sap or root pressure, is to restore the function of the xylem. Soil, it's a stop and go process. The temperature in the soil has to be above about 45 degrees Fahrenheit, or no root pressure can occur. The other function, in addition to pushing out air bubbles, is to rehydrate the buds. I've already talked about this. The water content in the bud increases from about 45 to 80% water just during that bud swell phase, and that is because root water is being pushed up through the plant. One problem, if you don't have enough soil moisture, which can be an issue, especially in the eastern part of Oregon and Washington, is that the plant can then not generate enough root pressure. This happens sometimes in our winters when we don't have enough winter rainfall and not enough moisture in the ground. And when we don't have irrigation water available in the spring, we can sometimes see erratic bud break, low shoot vigor, often a bit of a zigzag growth of the shoots. And we see clusters like here aborting. They fall off just as they come out from the bud. And we also see poor fruit set. That is often the result of insufficient soil moisture, insufficient root pressure, and insufficient sap flow. Typically, what I recommend is you can maximize early season growth, canopy development, and yield if you keep the soil moisture within about 3 to 4% of the field capacity of the soil, which means that you need to know your soil. You have to know what the permanent wilting point and the field capacity is because, as you can see in this picture up here in this graph on the upper left, that field capacity and permanent wilting point, or the water envelope or window of the soil, depends on the soil type. The sand cannot hold as much water as a loam, for example. So the common recommendation that I have, if you know that it's warm enough and you should be seeing sap flow, you should be seeing these drops of the pruning wounds, and you don't, irrigate, turn the water on, because chances are the soil is too dry and the vine cannot generate enough root pressure. Then we have bud break. The buds are now breaking. The temperature increases above 50 degrees Fahrenheit, which we call the base temperature of grapes. We tested that over here in an experiment with Julie Tarara, who many of you know in Oregon. We blew warmed air or chilled, cooled air on buds before bud break. And we can show that if we keep the bud over here below 50 degrees Fahrenheit, it does not break. If we heat the bud up, it breaks much faster. Shoot growth is much more rapid. Again, bud break and shoot growth are a temperature response. That base temperature varies. Vitis Bellandieri has a high base temperature, so late bud break. Vitis riparia, the northern grape, has a low base temperature, breaks very quickly when it gets warm in the spring. And vinifera is somewhere in between. The varieties, I'm not going to go through all of these varieties, also vary somewhat in their base temperature. We know that Riesling and Cabernet Sauvignon have later bud break, for example, than Chardonnay and Gewürztraminer, because the base temperature varies between varieties. But it's roughly around 50 degrees Fahrenheit. Cell division in the bud starts about 1 to 3 weeks before bud break, and this is how we see buds swell. The buds are swelling because their cells are dividing, they're pushing out of those bud scales. And then we have auxin, the growth hormone auxin, being formed by those swelling and breaking buds, which reactivates the cambium and produce new phloem and xylem in the trunk and in the cane, supporting the new shoots from growth, for their new growth. At the same time, this auxin has another function. On the upper left, you can see a Pinot Noir vineyard in Oregon, in the Willamette Valley in spring. My interpretation of this is that this is cane pruning, and these canes are being pruned, they're too long. Maybe the vine spacing is too big, or this vineyard should be converted to spur pruning. You can see this uneven, this wavy bud break, where you see lots of growth at the tip of the canes and essentially nothing at the base. This is a classic case of what we call correlative inhibition or apical inhibition. The auxin that is produced by these swelling buds travels down the cane and inhibits bud break at the base of the cane, but not in an adjacent spur. So this is just a within cane phenomenon, but not a within vine phenomenon. This is why I'm saying you can easily solve this problem. If this is a problem you have, you can easily solve it by converting from cane to spur pruning. You can also utilize this issue and delay bud break. If you're in a spring frost prone situation, spring frost is a problem at your site, you can then use this correlative inhibition or apical dominance or whatever you want to call it to your advantage to delay bud break. This is an experiment that I'm presenting from New Zealand. I've changed the months of pruning so that we can understand them a little better in the Northern Hemisphere. Vines are pruned in January, February, March, or April. And you can see very easily what effect that has on the time of bud break. So that means late spur pruning will delay bud break because essentially you keep the canes on the vine at the buds at the tip of the cane break, but not at the bottom of the cane. So that when you come back later, you then remove that inhibition, and finally the buds can now grow out, they can break. But later, you can delay bud break by up to 3 weeks approximately by pruning late. In practice, in our climate, we often pre-prune in winter to about 5 or 6 buds or canes in the winter. And then we come back and hand prune as close to bud break as possible, or if you're in a really frost-prone site, after bud break. So I'm supposed to talk a little bit about the basics of pruning, and the last few slides are just about pruning before we close this presentation. A few pruning goals. As you know, pruning determines the number of buds, the position of those buds on the plant. If you never prune your vine, this is a picture I took years ago in upstate New York. This is a wild grape. You can see what happens if you do not prune. These plants grow to very large size. They grow up on trees. They produce a huge amount of fruit. So pruning also serves to maintain the size and shape of the plant. And of course, it sets the upper limit on yield potential. And I'll show you a slide about that a little bit later. Just quickly, the golden rules of pruning. These are from Richard Smart. Many of you know Richard Smart, a consultant originally from New Zealand, but he's traveled in the US extensively. He's in Europe these days. He coined the term golden rules of winter pruning, but the concept itself goes back to Nelson Challis, who was a viticulturist in upstate New York and came up with this idea of balance pruning. Balance pruning means that you can weigh what you prune off. You can use a fish scale, like in this case, bundle up your prunings, take the weight of the prunings, and then you're supposed to leave about 14 or 15 buds for each pound of pruning weight. So the more vigorous a vine is, the bigger a vine, the more wood, the more buds you should leave behind in order to control that vigor somewhat. You should also leave enough buds to get about 5 shoots per foot of canopy. This 5 shoots varies a little. In Washington, we have up to 10 shoots per foot of canopy. So it very much depends on the extent of light that you get during the summer. In a climate with a lot of light, you need more shoots to provide a little bit of shading in the canopy. So this has a lot to do with canopy density. The third rule of pruning is to avoid cross row shading. This picture from Oregon shows a nice Smart Henry trellis. Looks beautiful up here in the front, but as soon as you go back into this row, where 2 rows start together or grow together, you can see that this row casts a shadow. There's a lot of shading on the other row that prevents that row or that portion of the row from producing good quality fruit. So avoid cross row shading is another rule of pruning, and this has a lot to do with planting density and vigor, but also with the number of buds that you should prune. And then, of course, you should apply all of these 3 rules simultaneously, which means that if you have to leave too many buds, chances are your vine is too vigorous. And you can take some of that vigor out by dividing the canopy, either Scott Henry or a lyre or some other divided canopy system, Geneva double curtain, whatever you prefer, to take vigor out of the plant. And here's an example of how this vine vigor bud relationship works. We did this experiment a few years ago where we pruned Merlot vines anywhere from 10 to about 130 or 140 buds per plant. So a huge variation in bud number. And you can tell when you look at these pictures or graphs up here, the vigor per shoot, the growth rate of each individual shoot decreases as the bud number increases. And of course, this is because the number of shoots increases as the bud number increases. At the same time, the amount of yield, of crop yield per plant increases as you have more shoots, but the size of each cluster decreases. You can see this down here in the pictures. This is on the left-hand side, a standard spur-pruned, a young Merlot vine, and then essentially an unpruned Merlot vine with a bigger fruit zone, more open and smaller fruit clusters. This is about the pea-size stage, what you can see here. So big difference in how vines respond depending how many buds you leave at pruning. So in very simple terms, with more buds, you get more shoots. With more shoots per vine, you get lower vigor per shoot. But you also get more clusters because every shoot potentially can have 2, maybe 3 clusters, right? as you have more clusters, you get higher yield, but more clusters or higher yield also means that that vine has more flowers. More flowers usually means lower fruit set. The vine constantly self-regulates and compensates. So as you have more flowers, you have lower fruit set, and because you still have more berries on the vine, those individual berries are also smaller. So you can use that bud to shoot relationship to gauge somewhat your yield level of the plant, but also the size of your berries, the amount of fruit set, the openness or compactness of your clusters. If you leave more crop early in the growing season, you will get less compact clusters overall. So this is a classic relationship of bud number and then vine compensation. Here's the last slide that I have for you today. And this should set you up for the next presentation by the Italian group that will talk more about sap flow and pruning rules and so on. One thing to keep in mind when you prune is that winter pruning, like anything that you do, you remove something from a plant, that is an injury to the plant. If you prune during dormancy, the plant feels injured, it has a wound, you have made a cut here, here, a cut with your pruning shear, and the plant responds even during dormancy. Metabolism, remember, continues during dormancy. It responds by producing a gum. It secretes that gum into the xylem of the cane that you have pruned off, and that gum is mixed with phenolics, which are antimicrobial, so it's a protection, a wound response, a protection of that pruning wound. The gum will seal the xylem here at that pruning site about a ½ an inch deep. That means you shouldn't cut too close to your bud that you want to retain, and certainly not too close to the cordon, or you will get these desiccation cones. This is a picture I just got actually about 2 weeks ago from an Italian group at the University of Turin that are using micro CT, microcomputer tomography. That is basically an X-ray imaging of plant parts, and then they can reconstruct these desiccation cones. This is an older plant, and all the brown material, you can see that cone up here on the right, offset to the side. This is the same cone as what you see here. That cone has now gradually made sap flow on that plant less and less effective, efficient. So the morals here is do not cut too close to existing wound, leave a little bit of material on the plant, not too close to the cordon, not too close to the trunk if you leave replacement spurs on your vine, if you do cane pruning, or you will get these desiccation cones that will impede sap flow. And this is all I have to say. The Simonic and Search people will talk to you much more about sap flow and about actual pruning rules after this presentation. Just very quickly, because I have a captive audience here, I've been preaching from my book. A lot of what I've been talking about was just published in the 3rd edition, just published in January 2020, actually, about a year ago. 3rd edition came out, so you can, if you want to know more about what goes on during dormancy or during the growing cycle, you can read this here in this edition, or if you prefer the Chinese edition, you can look it up in Chinese as well. Thank you. This is all I have for today, and hopefully you will enjoy the presentation. I will be there live while this presentation plays. Unfortunately, I will have to listen to my own presentation for a change, but I will be there to answer any questions you may have during the chatting session as well. Thank you. Goodbye.